want to talk to you in this video all about tracheal mite dissection. So tracheal mites, like the name implies, are mites that get into the tracheal system of the honeybee. And while they're not as much a problem as people once thought they were, you hear pockets of tracheal mites popping up all around the U.S., and for that matter, we know they have a distribution in many countries around the world. So I want to talk to you a little bit more definitively about how you would identify tracheal mite infestations in the field. So the first thing that you're going to need to do is you're going to need to collect bees from hives that are more likely to have tracheal mites than other bees from those same hives. It's real easy to think about. Essentially, older bees are more likely to have tracheal mites than younger bees. So when you go to a hive, you're really going to want to target those older bees, and there's a handful of ways that you can do that. First, you can block the entrance of the hive so that all the foragers that are coming back are unable to go into the hive, and now you've got this assortment of foragers hovering around the nest entrance, and with an insect net, you can just sweep across that nest entrance and you can have all of those bees ready for tracheal mite dissection. I don't typically do it that way myself. Usually when I want bees for tracheal mite dissection purposes, I'll actually go into the hive, keeping in mind that older bees tend to hang out on the outer frames or the uppermost boxes of the hive rather than in those center frames of the brood nest. So I would go down to the brood nest probably, and from the outer frames of that box, I would collect adult bees. Doing that ensures that you're getting bees that are more likely to have tracheal mites. One little final tidbit that might be useful for you to know is that heavily infested bees often crawl away from the nest on the ground around the hive. So if you believe you have a tracheal mite infestation, targeting those bees that are crawling on the ground around the hive is a really good thing to do because you'll increase your chances of finding tracheal mites should they be in the bees. So I've talked about where to find those bees at the nest entrance, on the outer frames, or on the ground around the colony. What do you do to collect them? That part is simple. You just need a small container. This is just a little plastic cup that we have around the lab. You can use glass jars. You can use any other number of containers that have a good sealable lid. And all we do with this container is we half fill this container with ethanol. So ethanol is the drinking alcohol. A lot of people don't have access to pure ethanol or high concentrations of ethanol like we do here in the lab. So don't worry, isopropyl alcohol or rubbing alcohol will work well. So you can half fill this container with rubbing alcohol. Then you can go to your colony and collect bees off the ground, get bees from that net, or go to those outer frames and rub that open container down the sides of the frames, and that will cause bees to fall into that isopropyl alcohol. Once you have enough bees, somewhere in the neighborhood of about 50 or so bees, you can return the lid to the container and take that container of bees with you back to wherever you're going to do the dissections. And I just happen to have a container with collected bees. You really want to do these dissections with fresh collected bees. You don't want to go outside the hive, see dead bees on the ground, and try to do the dissection with dead bees. The tissue's just too hard, it's too dry. Using freshly dead bees, bees that you euthanize yourself by putting them into isopropyl alcohol, ensures that you're going to have the highest quality specimen for that dissection. Now that you've got your bees in isopropyl alcohol, you're ready to do the dissection. Just a quick word of warning. You don't want to collect bees into water. I don't like that method of euthanizing bees. It's just not fast enough. It's really not fair to the bee. But secondly, having bees in water means that when you're working with those bees during the dissection process, you'll have a lot of bubbles forming around the bees, and it makes it very difficult to visualize. So alcohol is just the substrate you want to use. With these bees in hand, now you need some basic supplies and at least one piece of equipment. We'll start with the equipment. You need some sort of dissecting microscope. This is not the type of microscope you were likely to have looked at cells in, bacterial cells or other cells in when you were in high school. This is a dissecting microscope that's supposed to give you fine resolution of larger items, things that you want to just have a look at under the microscope. So we're not putting bees onto glass slides like you might have learned in grade school. Instead, we're wanting to look at the whole body of the bee, and a dissecting microscope like this that gives you 10, 20, 30, up to 50 magnification is a really good start. A lot of the dissecting microscopes today have both the eyepieces that you can look through while you're doing the dissection, or they contain these digitized pads where you can do the dissection from here and see it on the screen, making it more easy to do. Also, dissecting microscopes have their own light source, but you really need to be able to see what you are doing when you are inside the bee. So I like to have this additional light source. These are called gooseneck lights because it looks like a long neck of a goose with these heads or lights at the end. And having these gooseneck lights make it possible for you to put the light where you need it to be while you're looking through the microscope for dissection purposes. Now, admittedly, these can be expensive. They can be $500, $1,000, $2,000 or more. If that's a problem, you might consider talking to your local bee club into purchasing one of these dissecting microscopes so that it's available for you to use or for members to check out. It's a great resource to have, not just for dissecting bees for tracheal mite purposes, but also for just looking at how beautiful bees are in general. So now that you've got this microscope and a fancy gooseneck lighting system, everything else is actually pretty easy. I like to have an extra container of alcohol here. This happens to be ethanol, but again, you can use isopropyl or rubbing alcohol. You need 2 sets of forceps. These are needle nose or fine tipped forceps that you're really going to need for handling that bee while you're dissecting it. You're going to need some sort of substrate into which you secure the bee. Now, this happens to just be a glass petri dish. I work in a laboratory. We have lots of petri dishes here. But any shallow container, like the lid of a jar, would work well. And when I tip it this way, you're going to see that it's conveniently loaded with beeswax. We melted some beeswax, we put into here, and that beeswax allows us to push pins through the bee and secure those bees into that beeswax. It's a nice little cheat for you when you're developing something into which you can place bees. Now, I said the word pin, so in order to pin bees into the beeswax, you're going to need pins. These happen to be insect pins. If you've hung out or known any entomologists, we have insect pins that we use to preserve insects for insect collections. You can get pins of different thicknesses. I like to have a pretty average size, a standard size insect pin. You see 2 of them stuck into the wax here. The purpose of these insect pins is to secure the bee so that while you're dissecting it under the microscope, it's not moving or shifting so that you can control what it is you're actually trying to do. A little bit of a cheat code with insect pins and securing bees into that beeswax. If you use 1 pin through the bee, while you work that bee, it's going to pivot around that pin. That's why I like to use 2 pins that go into the bee to keep that bee from pivoting. So the cheat is this. If you take a standard pencil with an eraser, you can cut the top off of the insect pin and stick the pin into the eraser, point side up, and if you do that with 2 pins, you now have something with which you can use to push through the bee and secure it nicely. That pencil gives you a nice handle to push those pins into the bee rather than having to manipulate these fine pins. So we've got the bees, we've got extra alcohol, we've got a petri dish with beeswax in it, insect pins, spare insect pins, 2 pairs of forceps, a dissecting microscope, and an extra light source. Now let me describe for you the dissection process. We're dissecting for tracheal mites, and fortunately the name of this mite really helps you zero in on where you want to be. You want to be in the tracheal system of the bees. If you remember bee anatomy, bees have very robust tracheal system in their thoraces. That's the thorax. That's the part of the bee that everything that moves the bee attaches two. So think about a standard honeybee. You've got head, thorax, and abdomen. When you look at that thorax, you've got the 4 wings, you've got the 6 legs. So 6 legs and 4 wings are attached to the thorax. That means the thorax is a ball of muscle. It's muscle. It moves the wings, it helps move the legs. And what does muscle need? Well, muscle needs energy and it needs oxygen. That's why the tracheal system in the thorax is so robust. So what you'll get is you'll get tracheal mites entering openings at the side of the thorax of a bee. Those openings are called spiracles. Those are the breathing holes that line the side of the honeybee's body. The spiracles that occur at the base of the forewing, the forewing is the larger of the 2 wings on the side of the bee's body. The spiracle at the base of the forewings is large and leads to the largest tracheal system in the bee. So the tracheal mites will go through those spiracles and into the trachea of the thorax. And when you dissect a thorax, the tracheal system in it will look like upside down letter V's, with one of those legs of the V going to the side of the thorax, opening to the spiracle, with the other leg of the V going into the center part of the bee's thorax. So we are going to be dissecting that bee to try to get to that V-shaped tracheal system in the thorax. So now I'm going to walk you through the series of steps to do this. I grab an adult bee with 1 pair of forceps, and then with my hands I will position that bee such that I am holding that bee's wings with its belly facing towards me. And with the bee in that position, I use the 2nd pair of forceps to remove the bee's head and the bee's abdomen. You remove the head because it's that end of the thorax that has that robust tracheal system. So you need to remove the head so that you can get right down to the thorax. You remove the abdomen because I've found when I'm working with the bee under the microscope, if I accidentally press on the abdomen, it can cause that abdomen to release nectar or honey through the bee's digestive system and out the top of the thorax, making it difficult to see what I'm looking at. In other words, squeezing that abdomen can cloud my field of view when I'm working around the thorax. So I remove the bee's head and I remove the bee's abdomen. So what you should have left in your hand is you're holding the bee by its wings, the thorax is facing you, the legs of that thorax are up towards you. Now I will take a pair of forceps and place that bee's thorax into a petri dish on the beeswax. The petri dish I've already flooded with ethanol. So I'm not doing a dry dissection. The petri dish is flooded with ethanol or isopropyl alcohol so that when I put that thorax onto it, it is submerged completely under the liquid. It makes it easier to see things when you are working with that bee. Once that thorax is on its back, in other words, on its wings, I will take 2 insect pins and insert them from both sides of the thorax. Some people will pin the bee first and remove the head and the abdomen. Some people will remove the head and abdomen first and then pin the bee. In this case, the thorax is pinned using a pin on either side, which pins it belly up towards you. And again, you can use 2 individual pins, or you can use that pencil cheat I told you where you stick 2 pins into the eraser of a pencil, and now you've got an easier handle to use as you pin that thorax down. Now with the legs of the thorax looking up to you, this is the trickiest part. A bee's thorax is composed of 3 different parts: the prothorax, the mesothorax, and the metathorax. Each of those sections has its own pair of legs. So the first pair of legs is attached to a piece of exoskeleton that has to be removed. So the first thing I do is with forceps is I'll grab the front pair of legs and lightly and forcibly remove them from the thorax of the bee. So if you do this well, the front pair of legs comes off, a little bit of the exoskeleton in the middle part of that thorax comes off, and you're able to see muscle tissue into that opening of the thorax. And here's the trickiest part of all. With that opening available and that first pair of legs removed, the thorax has what we call a collar. It is a section of exoskeleton that rings the opening of the thorax that you can see. So with both pairs of tweezers, you are grabbing the base of that ring and slowly peeling that ring off both directions from the thorax. And what that does is it opens the thorax for greater visibility. Now, this ring of exoskeleton actually covers the spiracles that lead to that biggest tracheal system in the thorax. So you have to be sure to remove that exoskeleton from the side of the thorax. This is incredibly important because at low tracheal mite infestations, the mites will only be at those openings, at those spiracles on either side of the thorax. So if you're only looking at the tracheal system in the center of the thorax, you might miss mites that have just invaded the tip, the spiracles. So you want to remove that ring, remove that collar all the way down to the spiracle. Once you've done that, you have exposed those two upside down V-shaped tracheal tubes that are on either side of the bee. Now what? Well, it's really simple at this point. You've done all the hard work. Now you're simply looking for evidence of tracheal mites. What is that evidence? Well, when the tracheal tubes are infested with tracheal mites, you will see white glob or circular shaped structures in there. That's the mites or the young mites or the eggs that the mites lay. So you can see these tubes, these tracheal tubes that are filled with these globular structures. Some of it's white. It looks completely clogged in heavily infested situations, or it might only have one or two of those oval-shaped structures at the tips of the tracheal system near the spiracles. Once tracheal mites have been in there for any length of time, they cause feeding damage that can cause the bee's body to melanize the tracheal tubes. Melanization simply means that they turn black. So if you have tracheal tubes that have these white globs in them or this black melanization, then you know you've got a tracheal mite infestation. This is a picture of two tracheal tubes that are free of tracheal mites. This is a picture where one tracheal tube has both those white globular structures as well as the melanization damage, while the other tracheal tube is free of tracheal mites. So you can get a good side-by-side comparison of an infested tube and an uninfested tube. You want to use this process on 10 to 20 bees so that you can get an estimate of the percent infestation. In other words, if one bee is positive out of 10 bees, then it's a 10% infestation rate. If one bee is positive out of 20 bees, it's a 5% infestation rate. But usually 10 to 20 bees are sufficient to give you an indication of a rough infestation rate and helps you know if your colony is suffering from tracheal mites. I hope the tricks I've shared in this video will be helpful to you as you dissect bees to confirm presence or absence of tracheal mites in your colonies.